Hello dear learners, in our today's class we are going to start with the mode of antigen presentation by the MHC molecules. So in our last class we had talked about the details of the structures of MHC class I as well as MHC class II. In our today's class we have to learn that how do these classes of MHCs represent the particular antigenic peptide outside of the surface of cell so that the T helper or T cytotoxic cells can easily identify. There are two modes of antigen presentation, that is endogenous mode and the exogenous mode. The endogenous mode of antigen representation talks about when the antigen which has been formed is produced inside the cell itself and then it is then expressed with the help of MHC onto the surface. While the exogenous mode talks about when the antigen is coming from outside, it is broken down into peptides and then these peptides are expressed by the MHC on the cell surface. So endogenous mode is mainly performed by the MHC class I while the exogenous mode is performed by the MHC class II. The peptide in the endogenous mode is synthesized within the cell, while in the exogenous mode the peptide comes out of the cell. Or I can say that peptide comes from out of the cell. In the endogenous mode, the proteasomes are involved, but in the exogenous mode, the endosomes are involved. Now, what are the two organelles? We will also discuss them once we start learning about the pathway. Okay, so this is how your classical whole MHC1 representation looks like. So here is the step-by-step process of endogenous pathway. So we are going to understand that how the pathway is following. For this, first of all, we need to look into the cytoplasmic details and we have to zoom in onto this antigen. So let's zoom here. We already know that there are certain antigens which can stay inside the cell and which can replicate and then they can produce their own new antigens. So those antigens which are produced in the cell itself, these antigens are represented via the cell through the endogenous pathway. What will be the steps? The first step is the ubiquitination or the tagging of the antigen. This is your antigen molecule. The tagging of the antigen molecule with the ubiquitin proteins. Now ubiquitin proteins play a very important role in sending these into the proteasomes. Now there is one of the important enzyme also, that is ubiquitinase. It utilizes the ATP and allows this ubiquitin tag molecule to be sent into the proteasome. Now this is the proteasome. It typically is of the sedimentation coefficient of proteasome is typically around 26s. So this is the proteasome and it consists of 3 to 4 subunits. It consists of some of the enzymes which are very much efficient in proteolytic cleavage and they can even cleave the peptide bonds between 2 to 3 different amino acids at the same time. Means their main role is the antigen has to enter inside and the antigen has to be cleaved inside the proteasome. Now there is one additional information which you need to know. If the proteasome has to function for the immunity, then inside the chromosome 4 we had the MHC genes. These MHC genes then start coding for some of the special proteins which are LMP4, LMP7, and then there are certain non-MHC genes also which are located on some another chromosomes and they code for LMP10. So these LMP4, LMP7 and LMP10 proteins also get integrated into the proteasomes. Now these have the main function because when these get integrated into the proteasome becomes more efficient in cleaving this particular antigen. So after the antigen has been cleaved inside the proteasome, the antigenic polypeptides will be generated. So these are your antigenic polypeptides. Now the antigenic polypeptides have to be transferred into the rough endoplasmic reticulum. In the meanwhile time, before they are transported into the rough endoplasmic reticulum, the processing of whole of the MHC1 molecule inside the RER begins. We already know that there are three alpha subunits of the alpha chain, that is alpha 1, alpha 2 and alpha 3. From the very starting, they are the three of them are bound together. Now there are two certain proteins, which is ERP57 and then there is kelnexin. Till the time beta microglobulin is not attached to this alpha chain, the kelnexin and the ERP57 is helping the immature MHC to be stabilized. Very soon after this, the kelnexin part is removed and in the place of kelnexin, the beta 2 microglobulin is attached, while the ERP57 still remains to provide the stability. After that, so that these can get into the proper shape and orientation, there are certain helper molecules or helper chaperones which are referred to as stepazine. These stepases are actually providing the shape to this particular and the classical MHC1 molecule. Till the time ERP57 and calreticulin, the another new molecule, are attached to this MHC class I so that they can provide it its accurate shape and also let it stabilize in the environment. In the meanwhile time, the antigen, which was the antigenic peptide which had long transferred from the cytosol into the rough endoplasmic reticulum, is now to be attached in this place. But before that, how will it enter inside the RER? There are two important proteins here, TAP1 and TAP2 proteins, which have some of the hydrophobic regions which particularly anchor the membrane of the endoplasmic reticulum. Once they are allowing this to go inside, the antigenic peptide will enter into the RER, inside the RER matrix. What you need to know here is that these TAP1 and TAP2 proteins, which we are discussing about, have some specific things. Firstly, they will utilize ATP to generate, or we can say that the ATP hydrolysis will be done. Let me make it a little bit thinner, the pen tip. So they will do the ATP hydrolysis for transferring these particular molecules, means they will require the ATP so that they can transport this antigenic polypeptide. And secondly, when I talk about their selectivity, the selectivity of TAP1 and TAP2 proteins is only for about the peptide which is 8 to 10 amino acids long. That's why we know that in the cleft of MHC1, the antigen with 8 to 10 amino acid only fits. Now the peptide has already entered inside the rough endoplasmic reticulum, and now in the place of ERP57 protein and in the place of calreticulin, now this has to be bound. So as soon as the peptide comes here, the two of them are replaced and the two of them are removed, and the antigenic peptide comes and binds into the cleft which is formed by alpha 3 and alpha 2. Now this is what a classical MHC1 molecule with the antigenic peptide will look like. In this stage also, what we can notice is that it is not yet packaged. Means the alpha 3, alpha 2, alpha 1 as well as beta 2, the 4 subunits are there and also there is the antigenic peptide. But now this complex has to exit the rough endoplasmic reticulum. Once it exits the rough endoplasmic reticulum, there in the Golgi apparatus, when it enters, it has to undergo packaging. So this is how your mature MHC1 has to look like. It is packaged, it consists of alpha 3, alpha 2 and alpha 1 subunit which together comprise of the alpha chain, and there is the beta 2 microglobulin and these are attached by the non-covalent linkages. Then here is the peptide which has fit into the cleft which is made up by alpha 2 and alpha 3. Now this MHC1 is further transported outside onto the plasma membrane. As we already know, this is how the typical of this should look like. Also here this is completely packaged and there is this antigen which is present. Antigen, when I talk about antigen, I actually mean the peptide of that antigen, not the complete antigen. Now this cleaved peptide, which is represented by this MHC1, has to be recognized by the CD8 plus cells. So CD8 plus cells with their receptors, that is CD8 plus receptors, will then recognize this particular antigen, and accordingly then they will start their response, wherein they will either proliferate, or they will produce chemokines and cytokines, or they will also activate the B cells. So this is how your typical endogenous pathway goes. In the next class we are going to talk about the exogenous pathway, wherein we are going to talk about the MHC class II and their particular representation of the antigen.